Namaste, and welcome back to Typical Monk. Last class, we saw the structure of DNA. Today we see how one DNA becomes two, that is, replication. It is a beautiful semi-conservative process proved by a classic experiment. One idea at a time. Here is today's path. 5 stops. First, semi-conservative replication. Second, the Meselson-Stahl experiment. Third, the replication fork. Fourth, the enzymes of replication. And fifth, the leading and lagging strands. First, the big idea, semi-conservative. Each new DNA keeps one old strand and builds one new one. That is why it is called semi-conservative. The old strand acts as the template, base pairing does the rest. Watson and Crick predicted it. It beat the conservative and dispersive ideas. Now, the proof, the Meselson and Stahl experiment. They grew E. coli in heavy nitrogen 15, then shifted it to normal nitrogen 14. The DNA was separated by density gradient centrifugation in cesium chloride. After one generation, a single hybrid, that is intermediate band. After 2 generations, half light, half hybrid, exactly what semi-conservative predicts. Now, where it happens, the replication fork. Replication begins at a fixed spot, the origin of replication, that is ORI. Helicase unwinds the double helix, and a Y-shaped fork forms. Now, each separated strand waits to be copied. Now, the toolkit, the enzymes of replication. Helicase unwinds and separates the 2 strands. DNA-dependent DNA polymerase, the main enzyme, it adds nucleotides only in the 5' to 3' direction. Primase lays a short RNA primer to start, and the polymerase is very fast and accurate. Ligase seals the joins in the new strand. Now, 2 strands, 2 ways. Look at this picture. Because the polymerase works only 5' to 3', the 2 strands are copied differently. The leading strand is made continuously without stopping. The lagging strand is made in pieces. These are called Okazaki fragments. And then ligase stitches those fragments together. Now, the high-yield pointers, the facts that NEET turns into questions. 1. Replication is semi-conservative. Meselson and Stahl proved it in E. coli with nitrogen 15 and 14. 2. The main enzyme, DNA-dependent DNA polymerase. 3. The leading strand is continuous. The lagging strand is in pieces, that is Okazaki fragments. 4. Helicase unwinds, primase primes, ligase seals. 5. Taylor proved it semi-conservative in eukaryotes, in Visha Faba. Now the first drill, assertion and reason. Each time, ask, is it true? Is R true? And does R explain A? First, R, the lagging strand, is made in fragments. R, DNA polymerase can add nucleotides only in the 5' to 3' direction. Both are true and R explains A, so the answer is A. Second, a Meselson and Stahl proved conservative replication. R, they used nitrogen isotopes and density gradient centrifugation. Here A is false, but R is true. So the answer is D. Third, each daughter DNA has one old and one new strand. R, replication is semi-conservative. Both are true, and R explains A. So the answer is A. Now, the second drill, match the column. Make the correct pairing. Unwinds the helix, helicase. Adds nucleotides 5' to 3' DNA polymerase. Seals the fragments, ligase. And the semi-conservative proof, Meselson and Stahl. Now the third drill, how many of these statements are correct? Check them one by one. First, Okazaki fragments form on the lagging strand. This is correct. Second, DNA polymerase can build a strand 3' to 5'. This is wrong. Third, Meselson and Stahl work with E. coli. This is correct. So the correct statements, 2, that is, the first and the third. Now let's clear the mistakes students fall for. Myth: D and A polymerase can synthesize in both directions. Truth: only 5' to 3'. That is why the lagging strand is discontinuous. Myth: replication is conservative. Truth: it is semi-conservative, one old strand and one new. Now test yourself. Answer each one before it is revealed. Answer first. That is how you build strong NEET recall. Question 1. The type of D and A replication? Answer: semi-conservative. Question 2. Who proved it? In E. coli, Meselson and Stahl. Question 3. The direction of D and A polymerase? 5' to 3'. Question 4. The fragments on the lagging strand? Okazaki fragments. Question 5. Which enzyme seals the gaps? Ligase. Let's gather it all in one breath. Semi-conservative, one old strand, one new. Meselson and Stahl proved it. E. coli, nitrogen 15 to 14, cesium chloride gradient. Helicase unwinds, polymerase builds 5' to 3', primase primes, ligase seals. Leading continuous, lagging in Okazaki fragments. Taylor, semi-conservative in eukaryotes, in Bishifaba. In the next class, DNA to RNA, that is transcription. See you there. You have mastered how life copies itself. Be proud. One idea at a time, with a calm mind, and you will crack NEET. This is typical monk. Every class, always free.